All right. Thank you for coming to another HPL seminar. Today, we have a special guest visiting from the University of Waterloo, Jackie Zehr. Jackie did her undergraduate and master's degree at the University of Toronto, where she studied kinesiology. And she finished her master's degree project on in vivo spine biomechanics. She then moved to the University of Waterloo to work with Dr. Jack Callahan in 2017, and where she studies lumbar spine tissue and injury mechanics. She's currently a PhD candidate, expected to defend in April, May, sometime in the spring.
Yeah.
I won't say when. All right. Aside from that, Jackie does some teaching at the university, and she's an independent day trader, trading low float stocks. Yeah. Okay. And then her current research interests are in spine biomechanics, mechanobiology, cartilage mechanics, and tissue load management. She's interested in learning about cartilage mechanobiology, which is why she came to visit us this week. So, and aside from that, she's also a pilot, so she can fly her own private plane. So that's cool, like a former HPLer, Colin Furmanger. So she does have a Twitter handle too. If you want to ask her about anything, it's just Jackie Zehr, one word. Yeah. So I'll turn it over to Jackie to get things started for a presentation titled "Compression-Induced Changes to Spinal Joint and Endplate Mechanics."
Thank you, and thanks to Walter, Olivia, and Michael for having me. To start and provide just a little bit about my research path, and Michael alluded to a little bit of it. I did my undergraduate degree at the University of Toronto in kinesiology, and there I started working with Dr. Tyson Beach quite early in my undergraduate. I didn't do research right away, but my undergraduate thesis and project was a small project funded by Own the Podium, where I used some optimization to relate characteristics of anthropometric, the block configurations, and characteristics of force production in the national 100 meter and 200 meter men's and women's sprinters. I stayed in Dr. Beach's lab for my master's degree, and I deviated from sport biomechanics into spine biomechanics, which aligned with more of his program and expertise there. My thesis looked at if some of these common nonlinear characterization approaches are actually sensitive to some biomechanically distinct profiles of spine kinematics and kinetics during lifting tasks. I loved the technical aspects of the in vivo lab, and it's something I still really enjoy, but I developed more of an interest in some of the fundamental science and the tissue work that I had been reading about at the time, which also aligned with a job that I had in the Division of Anatomy at the University of Toronto at that time as well. And so after finishing my master's degree, I started my PhD right away with Dr. Jack Callahan at the University of Waterloo. And this is where I currently am. And here my work has largely focused on injury mechanics to the lumbar spine tissues. And this work will make up most, if not all, of the talk today. And so I mentioned that a major focus of this PhD work was studying low back injuries. And so I want to just first start by introducing this spectrum of magnitude-dependent compression injuries in the lumbar spine tissues. And of course, the lumbar spine experiences highly complex and multi-directional loads. But for this talk, I'll be focusing on compression, which is generally the axis, the primary axis of loading, and the axis in which the spine can withstand the most force. So at the low end of the spectrum, so this 0 to 30%, so yielding compression loads in the range of 0 to just over 3 kilonewtons. The intervertebral disc is largely susceptible to injury, especially when it's paired with dynamic and highly deviated lumbar spine postures. And this combination of low compression and spinal flexion stresses this posterior portion of the annulus fibrosus, which over time tears and provides this avenue for the nucleus pulposus, which was this purple blob, which was once in the middle of the intervertebral disc, to travel posteriorly and leave the disc, causing herniation and potentially nerve compression. Now, in the mid-range of this compression loading spectrum, so between 30 to 70% of the ultimate compression tolerance, and this yields loads in the range of 3 to about 7.5 kilonewtons. Previous work from our lab has demonstrated that the cartilage endplate will always fail, typically in the form of a fracture, before injury to the disc, or macroscopic injury at least to the disc, is observed. And these include fatigue fractures, which are shown by these two images here, so a stellate fracture and a step fracture shown on the bottom, as well as which is a different failure pattern, this Schmorl's node injury, which is commonly clinically defined as this vertical herniation. And we recently dissociated the mechanisms between the Schmorl's node and these fatigue fractures in the final study of my PhD that I just finished collecting. And then lastly, at this high range, so anywhere between 7.5 to about 12, 13 kilonewtons of compression, failure to the trabecular bone or burst fractures to the vertebrae themselves are commonly observed. And these types of injuries also coincide with generally a very high rate of compression loading. Now, historically, Jack's program has largely focused on the joint mechanics in this low range of loading, with a large focus on intervertebral disc mechanics and the pathways of annulus damage during prolonged sedentary postures, like office sitting and driving, as well as lower demand but highly dynamic movements. But a lot of the daily living tasks and occupational work that we perform actually falls within this mid-range of this loading spectrum. Is everything okay? Oh, sorry. So what I was saying is a lot of the occupational and daily living tasks that we perform actually fall within this mid-range of loading, making the end plate, so this piece of cartilage, susceptible to fatigue injury. And fatigue and damage to the end plate are highly debated as a starting point of this long and complex cascade of events leading to disc degeneration. And after reading how the end plate joint mechanics, how it affects the joint mechanics, and how it's very much understudied compared to the intervertebral disc, this mid-range, so this 30 to 70%, is largely where my doctoral thesis was positioned. And to give you just a little bit more information on this cartilage end plate, it's just this thin piece of cartilage that sits between the intervertebral disc and the vertebral body. And the end plate has two really important functions. It directly influences how load is transferred vertically through the spinal column. And it also has a profound effect on the physiological homeostasis of the intervertebral disc. So first, as I was mentioning, it aids in transmission of compression force through the deflection into the vertebral body in response to the pressurization of the incompressible fluid that is within the nucleus pulposus, which in effect offloads the radial stresses that are experienced by those inner layers of the annulus, which is likely why we see end plate injuries occur at these higher compression loads, as there's likely more vertical deflection in the cartilage. Second, this is a really porous structure, and since the intervertebral disc is avascular, this is the primary gateway for nutrients to enter the disc and waste products to leave the disc. And the ability for fluid to flow and macromolecules to diffuse through the pores of this cartilage is highly modulated not only by the health of this tissue, but as well as the characteristics of loading that it experiences. And although the end plate is categorized as hyaline cartilage, there are some very stark differences compared to the articular cartilage that covers the surfaces of our synovial joints in humans. So first, unlike articular cartilage, which is shown on the right, there's three distinct zones: the superficial, the middle, and the deep, where the chondrocytes as well as the collagen alignment have very distinct configurations between zones. In the end plate, the collagen fibers run in a single direction, that being horizontally, so parallel to that joint surface, as well as the cells also share that same horizontal configuration. Secondly, so as you'll see in this image down here, the end plate is really poorly anchored to the subchondral bone, unlike articular cartilage, where we see this embedding between the cartilage and the bone to add strength. And the end plate is really only anchored, as shown in these images, by these random collagen fibers, which makes it highly vulnerable to avulsion type injuries, where this cartilage becomes totally separated from the vertebral body. Now, with that background, my doctoral thesis had this global objective to understand some of the pathways and mechanisms of fatigue injury, as well as damage, as well as the associated morphologies of macroscopic and microscopic failure in this cartilage end plate. And so this is a flow chart of all the studies comprising my thesis. And we really approached this broad objective first with 3 studies that are on the left that were dedicated to the development and testing of methods. And those methods and some information that we gained from those studies were then used in the 5 studies that we conducted to collectively address that global objective. And so just to give you a little bit of an overview, so in study 1, I did a lot of comparative work between the porcine cervical spine, which we commonly use as a surrogate for the human lumbar spine, as well as some aged cadaveric human spines. And we were also really fortunate to have young donors below the age of 33 admitted into our anatomy program. And so we were actually able to extract the spine in one piece from those donors and include it in some side work that I have going on, as well as this comparative study. In studies 2 and 3, we examined how some of the mechanical properties, the collagen content, and the microstructural void development changed at various points throughout the lifespan of these of the spinal units. And the mechanical data gave rise to some mathematical characterizations that we made and published in study 5 on how this loading history, so the various histories that we implemented, influenced the trajectory of the ultimate compression tolerance throughout the lifespan. And then lastly, in study 4, which I just finished collecting right before Thanksgiving, we tried to elucidate some of the relationships between the compression paradigms that we applied in study 2, some of the microscopic damage that we observed in study 3, as well as some macroscopic and clinically relevant low back injuries. And so we specifically dissociated the fatigue fracture mechanism from this highly debated Schmerl's node. And this was exciting because we were first to reproduce the Schmerl's node in a laboratory macroscopically by experimentally manipulating the structural integrity of the subchondral bone localized underneath the central Endplate region. Now, I don't have time to talk about all 8 studies, but I'm excited to share some of the key findings that we found in studies 2, 3, and 5 specifically. Now, the study of mechanical fatigue and the progressive weakening of spinal tissues was really central component to my PhD work. And a really intuitive or common way to describe this notion is with a power law of cycles to failure, which is on the y-axis, and stress of the tissue, which is on the x-axis. And this function intuitively demonstrates that a higher stress is nonlinearly associated with fewer cycles to failure. And in the spine, in vitro data from the 80s has been used to develop exponential function based on some normalized compression data to predict the number of cycles that it can withstand until failure. And then from there, some element of risk is typically evaluated, usually where the cycle count sits on that function, or and more generally, the number of cycles to failure. But if we approach this problem of fatigue or overuse injuries from a more ergonomic, a load prescription or a load management lens, the margin of safety, so this difference between the applied load, shown in red, and the tissue tolerance, shown by this gray function, becomes a really important factor for not only evaluating risk, but trying to understand how much load can be safely tolerated. And we can have information on the applied load using musculoskeletal modeling techniques of various complexities. But in this diagram, this function that is commonly drawn to represent the tissue tolerance changes as a function of the previous loading cycles or someone's loading history is what we conceptually see in this diagram. And in the spine, and more specifically the end plate, there's very little known about the history dependence of the tissue tolerance or the mechanical properties, or how these factors change at different points throughout the lifespan. And so this knowledge really drove this first research question to understand the effect of different cyclic loading histories, specifically the effects of posture, the variation in the peak compression force, and the loading duration on mechanical properties like the ultimate compression tolerance and the post-loading indentation properties of the cartilage end plate. And we also examine the effect of these same cyclic loading histories on the collagen content and microstructural void development. So the study really went from more of a joint level response to studying the cartilage end plate independently to more of the microstructural and constitutive changes. And we focused on factors of posture, the variation in the pre-compression force, which I'll explain a little bit later on when I get to those experimental factors, and the duration, given their documented effects on the fatigue of spinal units. And for this study, 114 porcine cervical functional spine, so the 2 vertebrae, the intervertebral disc, and all of the joint spanning ligaments were included in the study. And I'll point out now that the experimental groups for this study are highlighted in gray, and a separate control group in its path is depicted by the blue coloring and arrows. So the joints were prepared by potting the spinal joint unit in these aluminum cups, which were attached to this rotation carriage that's interfaced with the materials testing system. And this rotation carriage is controlled by a separate torque motor to permit controlled rotation about the joint's flexion extension axis while simultaneously applying a compression load from the actuator of the load frame. And all spinal joints underwent a series of preconditioning tests, which included a static preload, which was used to offset the postmortem tissue swelling in the disc, followed by a range of motion test, which involves rotating the FSU about its flexion extension axis within its physiological limits to achieve this nonlinear moment angle relationship in both the flexed and the extension directions. We then fit this fourth order polynomial function to, so shown by this red line, to the raw data points that are shown by the blue dots. And the endplates of this linear region, the endpoints of this linear region, shown by the green dashed lines, define the boundaries of what we call the neutral zone. And I'll explain the postures and numbers that we take from this relationship when I get to those factors. But in identifying this neutral zone, first of all, or the zone of the mineral rotational stiffness, we can later use this information to normalize the postures between specimens, despite some potential differences in their lordotic geometries, which may affect that load moment angle relationship. And then after the preconditioning, one of 18 different combinations of cyclic loading were applied, each with an average normalized compression magnitude of 30% of the predicted ultimate compression tolerance. And I emphasize at this point, this ultimate compression tolerance is predicted using a regression equation that's been published. And this load magnitude, this 30% ultimate compression tolerance, represents the in vivo loading experience of a low to moderate demand lifting task. So the first factor we examined was posture, which had two levels of neutral and 300% of the flexion neutral zone. So neutral was simply the midpoint between the flexion extension boundaries of the neutral zone, and flexion was 3 times the distance between neutral and the flexion limit of the neutral zone. And this creates an extreme, but a physiologically achievable posture that would be achievable during a bending or a lifting task. So neutral was selected as this epidemiologically and biomechanically justified posture that's associated with reduced injury risk in the low back. And the 300% flexion posture was selected to represent a posture that's biomechanically linked to low back injury, so more high risk posture. Second, random variation in the peak compression force about that 30% normalized mean was a second factor, with magnitudes of 10%, 20%, and 40%, which were selected to cover the range of in vivo load variation during a moderately constrained lifting task that was presented by Van Dien et al. In 2001. So looking at this unitless signal on the bottom of the slide, the average peak compression here is simply 1, and it's depicted by the dashed horizontal line. And so the variation that I'm referring to is the magnitude that the peaks either exceed or do not reach the average of that line. So in this example, a maximum deviation of approximately 40% is present, given that the maximum peak that exceeds the mean is approximately 1.4 for an average of 1. Now, exploring this variation was important because load magnitude is nonlinearly associated in the spine with fatigue, as seen by these weighting factors that have been used to adjust the effective magnitude for fatigue failure risk. And we demonstrated in a separate and previous study how the random variation exposures to these higher compression magnitudes can reduce the fatigue lifespan that was withstood by functional spinal units. And we took this a step further and equalized the cumulative load, so the fourth time integral of each cycle that the spinal joints were exposed to. So this graph at the bottom demonstrates the integrated sum of the compression paradigms for the 10, 20, and the 40% variation groups. And because the lines are effectively on top of each other and they're actually equalized after every 5-cycle block, we exploited the effect of this variation only, and not that one group may have been exposed to more cumulative compression loading than the other. And then the last factor was loading duration, which had 1,000, 3,000, or 5,000 cycles, which brought the FSU to about 94% of its predicted lifespan. So we didn't want to take it to failure. We just wanted to try and get it as close as possible. And we estimated this from previous work on cyclic loading in Jack's lab. And that might seem like very few cycles, but given the loading frequency that we selected to offset the effect of the different loading rates in our variation groups, this is effectively an accelerated injury model. And then after the cyclic loading exposure, the FSU was transected in half through the intervertebral disc, and one vertebrae underwent some destructive ultimate compression testing, and the end plate of the other vertebrae was dissected off the vertebral body for indentation and some subsequent immunofluorescence staining. Now, because we wanted to examine the material and some of the micro damage properties, as well as that destructive joint tolerance, we had an indenter fabricated to the size and shape of the intervertebral disc. And Before conducting the experiment, we of course validated that we got similar stiffness, yield, and tolerance values with this indenter as we would if doing ultimate testing with an intact spinal unit, which is how it has been done in the past. And we applied an axial compression force at a rate of 3 kilonewtons per second until failure occurred. And note that because this is a purely vertical force in this test only, the factor of posture only applied to this cyclic testing exposure. And then for the indentation test, myself and a friend and colleague from the lab, Jeff Barrett, took on the challenge of programming the position and force control in this Modeman robot to do a few tests that we wanted to do for our thesis projects. And I think we both quickly realized that this was very much overkill for what we wanted and needed to do. And it took us very much longer than we expected, but we have it going, and it's now a useful tool in the lab for other tissue testing at loads that are higher than our micro tissue testing system, but much lower than we can apply in the instrument. So the indentation test then first involved using the width and depth dimensions to size normalize the central, anterior, posterior, and lateral regions. And then these corner pieces were used to mount the end plate on top of these aluminum stanchions. So we applied 3 trapezoidal indentation tests in each region using the Modeman robot. We applied at a displacement rate of 0.1 millimeters per second until that 10 Newton load was achieved, which was held for 30 seconds. And then we unloaded the specimen at the same displacement rate. So we gained quite a bit of information from these force deflection curves, but I'll be focusing on the stiffness of the loading phase, specifically in this central region, which is structurally the thinnest and has been demonstrated to be the most susceptible to injury. So we tested all of them, but I'll be focusing on the central for today. Now for some of the results. So starting with the ultimate compression tolerance, there was a 3-way interaction effect. So the data are separated with flex postures on the left and the neutral postures on the right. And then within each of these groups, we have each loading duration on the x-axis. And then within each of those groups will be the loading variation groups that we examine. And then this black bar under the loading duration of 0 represents the average of our control group. So starting with flex postures, the average compression tolerance after 1,000, 3,000, and 5,000 cycles was significantly less than the control, indicated by the asterisks. And these durations significantly differed from each other, indicated by the different letters. But within each of these durations, we didn't see any effect of the variation within this flex posture. Now switching our attention to neutral, the average compression tolerance after the 3,000 and the 5,000 cycle groups was significantly less than the control, again, indicated by the asterisks. But the average compression tolerance also significantly differed between these durations, indicated by the different letters. But an additional finding that we did see in neutral was the effect of variation, specifically after these 3,000 and the 5,000 cycle durations. So after 3,000 cycles, the 40% group resulted in a significantly less compression tolerance compared to the 10% and 20% group. And after 5,000 cycles, we had even more separation, where the 20% group now significantly differed from both the 10 and the 40% groups. And then lastly, to compare each of these durations between within each of the durations between postures. So we're comparing red to red, blue to blue, and green to green between the graphs. The compression tolerance was significantly less when cyclically loaded in a flex posture compared to neutral at all of these durations. Now, interestingly, the indentation stiffness of the central region mimicked these findings that we saw for the ultimate compression tolerance. So we observed the same 3-way interaction effect in similar pairwise comparisons. So I won't unpack the same interaction effect again, but to understand what might be driving some of these mechanical changes, we looked at some of our immunofluorescence results that indicated changes to the collagen content as well as some microstructural void development. But to first summarize these mechanical findings and some of the mechanism that might be driving the changes that we saw, we found that posture during cyclic loading, as well as the variation in that peak compression force within a neutral posture, both notably influenced the compression tolerance as well as the indentation stiffness. And a potential reason for this is the poroelastic response of the joint to elevations in intradiscal pressure that are associated with flex postures, as well as higher compression magnitudes like what would have been experienced by those variation groups. And to give you an example, this Schultz paper linearly shows how the pressure increases as a function of the moment to increase the magnitude of flexion. So So this elevated intradiscal pressure is associated with a lower permeability for fluid flow through the cartilage end plate. And there's evidence from a poromechanical finite element model demonstrated that this reduction in permeability is also associated with an increase in vertical deflection, specifically within that central end plate region. And so continuous exposure to higher deflection magnitudes may in effect accelerate fatigue and potentially reduce the tolerance to load. So we have not tested this mechanism. We have not gone further with it, but this is a potential theoretical explanation for it. Now, before showing the results for the staining analysis that I just mentioned, I want to bring some of this mechanical data and specifically that joint tolerance into an applied or more of an ergonomic lens. And to do so, I want to first come back to this diagram. As I mentioned, we typically draw this nice curve or function in our conceptual diagrams of overuse low back injury, but we actually lack numerical representation of the compression tolerance beyond the first or the initial data points, which is how much of the existing data from the 80s was obtained. And so with the compression tolerance beyond these initial loading cycles being largely unknown, and how it relates to low back injury, as well as how we use this information to assess low back injury, looks more like this. And this is problematic because in that mechanical data that I just showed you, we saw that what we did 100, 1,000, and even 5,000 cycles ago is likely not only to affect this tolerance trajectory, but it's likely to affect our low back injury risk because a cycle that we saw that starts at a moderate 30% is no longer a 30% force after so many cycles. And so using some of this ultimate compression data, we started with some just simple curve fitting analysis as our first step. And let me preface this with that this work is still very much largely ongoing, and this is just a very first step that still requires some validation. But on the left, for the flex postures, a single second order polynomial was developed given that there was no effect of the variation we saw in study 2. And then on the right, so in neutral, separate polynomials were made for each of the variation groups because we saw an effective variation there. Now, so just visually, we don't have any quantitative data to put to this, but just visually, the conceptual diagram of overuse injury, some compression paradigms yielded a fairly similar shape like we saw in the 10% and the 20% groups, but others, like inflection, did not. And so if anything, these initial functions demonstrate that the tolerance trajectory is quite sensitive to the factors of one's loading history. And currently, we're using some weighting functions that were developed in Jack's lab to try and extend these functions throughout a wider range of the compression load magnitudes. And again, with some additional validation, we hope we can incorporate functions like this into some existing ergonomic softwares that are typically used to evaluate and manage loads in industrial contexts. And to give you an example of one existing software is called LIFT, so the Lifting Fatigue Failure Tool. And it was developed by Dr. Gallagher and his colleagues from Auburn University. And essentially, this software uses a very simple 2D rigid link model of the spine to quantify or get an estimate of the joint contact force that's experienced by the L5-S1 joint based on a lever arm and a handheld mass that can be input by a user in the field. And this is a very simplified approach, yes, because these inputs need to be easily accessible by a practitioner in the field. But there's some modeling limitations that we hope we can help with as well. But the estimate of joint contact force is currently normalized to a very static value, ultimate compression, that they report in their 2017 paper. And then from there, they use these exponential equations to derive the predicted lifespan and damage estimates based on the number of repetitions performed. So like in that general power law fatigue model that I showed earlier and I presented at the start, damage here is highly dominated by the higher loading cycles despite being performed many fewer times. So now based on how this and many other ergonomic softwares work, there are two main impacts that we hope some of our data can have on these risk assessment tools and how they're used in practice. So first, we want to try and include a history-dependent ultimate compression function in replacement of these static values that are representative of the tissue in its healthy and unloaded state. And I know this gets really complicated with aspects of rest and recovery, but that's a long-term direction that I hope to pursue in my future research. And second is to encourage and promote the relevance of examining the margin of safety, in addition to the cycles of failure. So as the joint tolerance changes, and so can manageable load magnitudes, they can also be adjusted accordingly. And it's possible to very explicitly evaluate potential risk associated with all loading exposures, not just the loading, the handheld load that's input by the user. And we currently position this work largely within ergonomics, given the emphasis of ergonomics and occupational biomechanics at Waterloo. But load management, I'm sure many of you know, is a really hot topic in high performance sport, as well as occupational athletes like military and emergency response personnel. And this work may complement a lot of that discussion on how workloads and athletes that are exposed to and how they might affect their injury risk, their recovery, and ultimately what they're usually most concerned about is performance. Now, to circle back from this conversation on the mechanical data and what we did and are doing with it, recall that I mentioned after this indentation test, we did some immunofluorescence staining with the objective to look at how those compression paradigms that I explained earlier affected the microstructural damage and the collagen content in the end plate, as well as the subchondral bone. Now, to clarify the intention of this procedure, and our focus on collagen content was largely driven by some structure function data that was published by Dr. Lotz's group from the University of California in San Francisco. And their findings demonstrated a really strong relationship between collagen content and the tensile modulus of the end plate cartilage, and weak relationships between the proteoglycan and water content with the tensile modulus. And so given that strong relationship between collagen and tensile modulus, we focused on type 1 and type 2 collagen in this analysis. And in our study, the end plate tissue was stained in the transverse, so looking down at the end plate and the sagittal planes for collagen 1 and collagen 2 using a multicolor procedure. And I'll only be presenting results from the sagittal plane because it, for the most part, encompasses both the changes in collagen content as well as the microstructural changes that we observed. So in total, I stained over 200 slides for this study, and we got about 684 images from this study. And there are two outcomes that I'll focus on for this presentation, and specifically in the sagittal plane. And the first is the fluorescence intensity. So the density of the collagen proteins, which with collagen 1, which is primarily in the subchondral bone shown in green, and collagen 2, which is primarily in the hyaline cartilage shown by orange. So the brighter these channels, the greater density of the collagen proteins. And then as the collagen becomes damaged or cleaved, the primary antibody can no longer bind, and therefore the channels will become less bright. I'll also present data on void development along this border between the cartilage surface and the subchondral bone. And then the second analysis that we did was to quantify the prevalence of some very specific patterns of microstructural failure or microstructural damage within our samples that we examined. So if you recall, we saw a three-way interaction for the ultimate compression and the indentation stiffness data. But here there were main effects for duration, which is on the top, and posture, which is on the bottom. And I'll first point out that the numbers on top of these images represent the fluorescence intensity for each of these channels. And these values are expressed relative to our control group, which was given a value of 1. So starting with duration, there were no changes to the collagen 1 intensity, so the subchondral bone, compared to our control group, but there was a significant reduction in the collagen 2 intensity after 3,000 cycles, as generally seen by the gradual reduction in the brightness of that orange cartilage surface as duration increases. And then for posture, a greater reduction in collagen 2 intensity was observed for the flex postures compared to neutral, again indicated by a less bright cartilage surface. And to take this a step further, because if you recall, we had some early changes in that mechanical data. So we had some reductions in ultimate compression tolerance as well as indentation stiffness after 1,000 cycles, especially in a flex posture. But these findings weren't totally explained by the collagen fluorescence. And so we examined some subsurface void development where the three-way interaction effect showed up again. So for the flex postures down on the bottom, we did see an early and then a more systematic increase in the void area as a function of duration, which may have induced an early structural weakness. And then in neutral, the accumulation of voids occurred after 3,000 cycles and was limited to the 20% and the 40% groups. So to summarize this portion of the analysis, we found that some early changes to the mechanical properties may be attributed to subsurface void development, followed by a change to collagen 2 content. We didn't image or do this real time, so we don't know exactly. And the primary pathway seems to be same for flexed and neutral postures, only accelerated in the flexion postures. Now, this outcome has been, I guess, generally supported by some similar findings that have been observed in bovine articular cartilage explants that were exposed to a cyclic indentation test followed by some stress relaxation testing as well. And then for the second part of our analysis, for these microinjury patterns, we quantified the prevalence of circumferential pore damage in the transverse plane, as well as node avulsion and cartilage-only fractures in the sagittal plane images. And we used simple chi-squared statistics to compare the distributions of each of these injury patterns between vertebrae, spinal levels, and postures, and the conditions in each are listed here. And before I advance on to the next few slides, I want to draw your attention to these 2 images because they are the controls. And on the next slides, I'll show patterns of damage and how they progress from this healthy state. So this is an unloaded state. So in the transverse plane, so looking down at the cartilage surface now, the incidence of this circumferential pore damage was about 37%, and 57% of those injuries occurred in flex postures and 43% occurred in neutral. And there was no significant difference in the distributions between vertebrae and spinal levels. So compared to the 1,000 cycles, or that image of the control that I showed on the last slide, the collagen 2 around the pores is less, revealing more collagen 1 or the subchondral bone underneath. And this is likely because as the endplate deflects, those pores may compact and act as a stress riser, which might be why we see that collagen 2 disappearing around the pores, and then it becomes exacerbated towards the end of the lifespan. So we also saw some distribution differences in the sagittal plane, specifically between postures. So first, in a neutral posture, 82% of the cartilage-only injuries were observed. And the distinction here, compared to what I'll show you in the flex postures, is that you can see that the structural or the fractures are primarily limited to this cartilage surface with no real microstructural damage observed in the subchondral bone. But in flexion, we had 90% of nodes and 100% of the avulsion injuries were observed. So node injuries are characterized by the extension of this damage into the subchondral bone and the eventual penetration of the damage through the cartilage surface. And now the mechanism of the nodes and these microscopic nodes and the more macroscopic injuries is really contentiously debated, but this notion of subsurface void damage informed in a way how we probed that mechanism in that macroscopic Schmerl's node study that I told you about at the beginning. And then at the bottom here for these avulsion injuries to where the subchondral bone, we saw a significant effect of vertebrae here. So these injuries were more common in the caudal vertebrae of a spinal unit, so the bottom vertebrae. And we think this is because when you flex a joint, the anterior portion is in compression and the posterior point aspect will experience tension. And we think this tension aspect may cause the disc to pull up the cartilage endplate given its weak structural reinforcement, which may initiate this type of injury and propagate it via mode 1 failure. Again, we haven't tested that mechanism right now. This is just a hypothesis to build off of. Now, a main conclusion that we made from this work was that posture did not really appreciably affect the overall incidence of injury, but it did affect the pattern of microinjury that we observed. And we saw in different images representing loading durations that the size and of damage was more or less linearly and strongly associated with loading duration. And to bring some of this data back to the global objective that I set out to answer when I proposed this thesis, compression tolerance and indentation stiffness of the central endplate were significantly affected by posture, and this is likely a result of the altered load distributions that are experienced by this loading surface in these deviated postures, and there's extensive data by Adams and colleagues on that relationship. The variation in the peak compression force influenced the compression tolerance as well as the indentation stiffness, but only beyond the midpoint of that fatigue lifespan, indicating that in a practical setting, control of the peak loading exposures or abrupt exposure to a higher demand task may become particularly relevant beyond someone's acute, the midpoint of their acute joint lifespan. And then the underlying reason for some of these initial changes that we believe in the compression tolerance and the indentation stiffness, we attributed those to first the development of those subsurface voids, followed by a change to the collagen content. And then in terms of future directions, in my PhD, it felt like I really just scratched the surface of understanding some cartilage mechanics. And this is an area that I could and want to learn more about. Specifically, I really want to learn more about the rest and recovery responses to repeated loading. We made some initial steps to understand this part of the curve, but of course we rest and recover. And so we're interested in understanding how that rest and recovery may affect the recovery of those mechanical properties. And it's my hope, and as well as I also am very interested in studying the chondrocyte mechanics and some mechanisms of mechanotransduction in these cartilage surfaces. So my hope is that by spending some time dedicated to the study of mechanobiology and cartilage, I can integrate some of that knowledge into my future research that would experimentally aim to probe some of these mechanical mechanisms of degeneration and damage in cartilage surfaces of the spine, with the long-term goal to establish some models of maladaptation and adaptation to improve those existing criteria that we have for fatigue injury. And then to end with some acknowledgements, of course, this work wouldn't have been possible without my PhD supervisor, Dr. Jack Callahan, who's been an exceptional mentor to me both professionally and personally, and it's really been a privilege to work with and learn from him. I also acknowledge Dr. Tyson Beach, who was not only my master's supervisor, but he's been someone who has greatly inspired me and supported me ever since my first biomechanics class as a young undergraduate student. And he's someone that I still very much look up to, and I'm thankful for his mentorship. And as for fairly recently, it's nice to have him back at the University of Waterloo. And then lastly, Dr. Quadrilatero is a professor at the University of Waterloo who has been very generous with his time and has taught me what I know so far about staining analysis. And I'm really thankful for his guidance and his generosity to allow me to complete all that analysis in his lab and using his equipment. So, of course, there are many others who contributed to this work, as well as funding sources. So that's all the content that I have for today, but I'm happy to have a discussion.
Wants to start us off?
I'm wondering, I'm trying to wrap my mind around the collagen content in the slides that you showed.
Yeah.
Because in terms of collagen content, like it's actually the collagen is moving away from this region, it's pulled away from this region, but there is no loss, there is no loss of collagen in the sample.
So maybe a loss of collagen maybe wasn't the best way. So maybe in terms of like degeneration or damage to the collagen would have been a better way to phrase that.
Even the damage to the collagen, because the antibody, like, what does it recognize? Does it recognize like region? Even if it's a cleave, the antibody is still recognize the regions of the collagen.
Yeah.
So how? Yeah, even if it's a cleave, I don't see it decreasing the amount.
Yeah, so my understanding is that it binds to, it's based on the genetic genes of the collagen protein. And as those change due to mechanical damage, and Thermo Fisher had demonstrated that this was effective for understanding the thermal denaturation of collagen. And so we applied it to a mechanical state and had similar outcomes.
Hi, thanks for the talk. I had a question about your load variation results in the neutral and flex. So did you show that compression tolerance is more sensitive to load variation in the flex posture?
No. So I think our interpretation of this is that compression tolerance would be more sensitive to flexion than it is to variation in the compression force. And we believe that this is due to the poromechanical response of the joint. So we believe that flexion has a greater effect on that permeability than the smaller changes that would have been observed in those variation groups.
Okay, great, thank you.
Oh, wonderful talk, thank you. I'm really struggling to understand the variation results,
Sure.
like the mechanism of the variance there. And so either the cumulative load based on the weighting factor is wrong, and the variation is introducing additional load, or there is something about the time history of going from a smaller load level to a big jump and then back down with the variance. And so assuming that it is the latter, it's like the time history, what could it be? I mean, if the cumulative load's the same, what could it be about the introducing the variance that is causing the accelerated damage?
So I think this also gets back to some of those poromechanical analysis of the joint response to load, and that poromechanical model that I showed demonstrated that the higher compression also is associated with a reduction in permeability. So those cycles would theoretically be associated with greater deflection in that central region, which may, when experienced cumulatively, may cause accelerated damage. But this isn't, according to our data, this isn't seen right away.
Can I follow up on that? Because I was intrigued by that result as well. And I was wondering whether or not, you know, you have the variability, you have the low lows and the lows that are above. And I was wondering if the primary damage just occurs in the 40% high, just in the high loading cycles. I was wondering if you tried to exclude that by doing tests where you didn't do as many loading cycles, but you did less, but just always mimicked the high loading cycles. If that would give you a similar damage. So in other words, the ones that are 20% or 40% lower, they don't really do anything, but it's all the ones that are higher compared to average. They give you the damage, and so it's not really the variability, but it's the peak load that every once in a while you apply.
Yeah, we haven't explicitly done additional tests to test that, but that's in a way what we are theorizing as well, that it's the peak load magnitude that, according to some nonlinear weighting factors, is weighted with a much higher factor that's associated with risk compared to those lower cycles. And because that relationship is nonlinear, exposure to those higher cycles is not necessarily offset by exposure to the lower cycles.
I would, just intuitively, not knowing anything about spines, that would be my kind of how I would think about it probably.
Yeah, it's a question that we're grappling as well. Yeah.
Do you have any baseline stiffness measurements in all your different samples? One thing I'm just kind of wondering is, like, all these are probably out of different adapters, out of the same region. They're all from different bodies. So what was the baseline health every sample before you started your loading?
Yes, sorry, maybe I didn't make it clear. So this was on a porcine model. And so these are acquired from an abattoir. So the age and the health is controlled. So they're in a young, healthy initial state.
But we've even seen in like porcine knees, not of healthy animals, the quality of the cartilage isn't the same.
Yeah.
The quality of the bone isn't the same. So I was just wondering if you could possibly get any quick stiffness measurements before you started your testing to see if they were all roughly the same stiffness. So I'm wondering if some of the damage you're seeing could be pre-existing just from variability.
It's a good point, and we didn't do that. We don't have a way. So in the disc, it's a lot more. The research is a lot more developed. So there's a way to quantify the health. Like there's a scale where we don't have that currently in the cartilage. So that's an excellent point you raised. That could be a future another way to verify that initial condition. Thank you.
Sorry, I thought the 0 was the initial condition. It's not.
So 0 is no loading cycles.
But wouldn't that be kind of
that's the what we're calling the healthy state? But
initial condition that Andrew was referring to, that the loading cycle, or
is that what you're referring to?
Well, it's a different animal, right? Like it's a different sample. Like
it's a different sample of 6.
Yeah. No, that's just kind of what I was wondering. Is like, assuming each one of those is a sample group.
Yeah.
So it's a separate vertebrate group for every controls are just one.
Mm-hmm.
Okay. And so are these standard deviation or standard errors?
Deviation.
deviations. So it's fairly tight for
Yeah.
samples.
I don't know the lift app or whatever very well, the Sean Gallagher's thing.
Yep.
But you want to introduce some time history maybe into the thing. But I was just wondering how good is it at predicting injury? Like, is it good enough? And if you introduce this sort of time history dependence, I mean, would it get better? I'm just wondering do you know anything about how well it predicts injury?
Yeah, so I think they did a validation study in their 2017 paper. So I think their predictions, I don't know if they've done longitudinal studies, but the prediction that they're using is they validated their model against the data set that they used to derive the exponential. So that data set from the 80s by Brinkman et al. And so I think the limitation that we're hoping to try and address is that right now, only the risk associated with the handheld load that they input can be assessed. Where we're interested in understanding trying to create the ability to evaluate the margin of safety. So the difference between a load, whatever that is, and some estimate of a margin of safety, or sorry, an ultimate tolerance for a given history.
Yeah. I'm just wondering if anybody's tried to sort of use it to prospectively evaluate lower back injury risk.
So I don't think there's an epidemiological, like a longitudinal study.
Yeah.
Sorry, no.
Yeah.
Sorry, I misunderstood.
I have a question, Jack.
Sure.
Can you explain how you normalize the cumulative load between the variability conditions? Like
Oh,
how you're
sure.
accounting for the differences in loading rate?
Do you mean this?
Yeah.
Graph? Sure. So this is the integrated sum, so the integration of each cycle. And so effectively, we tried to create, we picked the cycle, the variation that would create the least damage between those groups. And then the cumulative damage the cumulative load was equalized. So it was the exact same after every 5 cycle block. So after every 5, so it might have differed, which it shows in this line that their lines aren't perfectly on top of each other. So there may have been some differences within that 5 cycle block, but after that 5 cycle block, it was bang on.
So how do you do that between every 5 cycle block? Like what's going on? So you're manipulating the characteristics like the loading rate of the waveform, not only the magnitude to equalize the integral.
Right. So this didn't account for anything with loading rate. It was just simply that integral of the loading waveform.
So the integral of the force time curve.
Yeah.
But you are weighting the magnitude.
Yes.
Yeah.
With those factors. Yeah.
Do you in bone it seems that load sequencing can matter, like going from low magnitude to high magnitude or high magnitude to low magnitude, not like this, the kind of variability. Do you think the same thing exists here? Like where if you did all 20%, let's say everything at 1 and then a whole bunch of 1.4 or the reverse 1.4 and 1.
Yeah, so there isn't, there's a current PhD student of Jack's, Mamiko Noguchi, has done some load sequencing work, and some in vivo work published by Sean Gallagher's group demonstrated that this, and they got at this analogy or this question with using job rotation as so someone who did a low job followed by high by low, and their findings were basically that if you're exposed to these high demand exposures, you're at a higher risk of getting injured. They didn't really see an effect of the exposure or the sequencing in vivo. So the results aren't out yet for the in vitro sequencing, but in vivo, there didn't seem to be an effect.
I love the variation finding because I think if you talk to your everyday kinesiologist, they will say variation is good. And you need variation, and it's going to reduce your risk of injury because you're not going to be loading the same thing the same way. And I guess this is a little bit different kind of variation because it's
kinetic.
It's all in the same. Yeah, it's kinetic variation.
This is not variation. This is
It is.
exactly the same load loading, just different load magnitudes, but I think,
You think,
Because
so
it's
what
all
you think is
compression.
that fourth curve being low, that's just, there you go, thanks. At 0.6, it's not gonna induce any damage. But the one that goes high, 1.3, is the one that induces the hit.
that would be my prediction.
That would be like for sure, that tall load is going to create more damage than the smaller load. But cyclically, that smaller load has to induce some.
Maybe, but I'd be really curious to do that experiment. I love to do this experiment, but leave these out, just do that one, that one, that one, that one, that one. All the ones that are higher,
Yeah,
and then compare it to the whole history.
yeah.
And then you would see what is actually due to the high ones and what might be due to the low ones. And my hunch would be they would be very, very different.
Is there an induced
So-
limit for
I don't know.
spinal units?
Like,
Is there a magnitude about which you won't see any damage?
so below, so there is, the weighting factor increases by a factor that's greater than 1 after 37.5%. So these cycles create a load that is now going to be, those higher demand cycles would create a load that is weighted now with a factor that's higher than 1.
And would that smaller fourth cycle be sort of below where you start seeing the weighting effect?
No, because
Okay.
if you recall, this average, so this is our average peak cycle, was 30%. So we intentionally chose a load magnitude on that border that we would just, those higher cycles would creep up to those to get weighted higher.
And of course, this is all done in force control.
Correct.
Do you measure creep?
Yes. And the height loss, we published that in the, actually, no, not in this paper, sorry.
But we quantify it.
Yes. And the height loss, so we developed a paper, a 2019 paper on just this variation method. And in that, the higher variation was more height loss as well. Just within the total FSU, we don't know if that's in the disc or where that is. It's just the total height loss experienced by the functional spinal unit.
Do you think it could reasonably be somewhere else outside of the disc?
I don't think so, because we're at the load magnitude where the end plate is susceptible to injury and not the disc. But we think oh, sorry, do you mean the height loss?
Yes.
Oh, sorry. No, we think it's the disc because that's where the fluid is.
Exactly.
Yeah. Sorry, I misunderstood.
You said at the beginning of the talk that most of the force is in compression,
Mhm.
but you have a quadruped animal.
Mhm.
And first principles, I would have thought most of the force was a sort of reflection issue. And if you went from a quadruped to a biped, do we know if the disc is structurally the same in a quadruped versus a biped, or does the biped meet more forces and therefore have some different structure to the disc?
Yeah, so it's a good point. So I'll start by saying that we use the porcine cervical spine as this lumbar spine surrogate because it's cantilevered off of the shoulders like our lumbar spine is off of our pelvis. And so when those muscles contract to extend the head, it creates lordosis in those lower cervical joints, which is what we see in the lumbar spine. Jack has done a lot of comparative work in his work that has shown the similarity in functional and morphological characteristics of the porcine joints compared to the human lumbar. Yeah.
Okay, back to the
Sure.
back to the issue of permeability.
Okay.
I'm not even thinking about the end plate, so I don't really know much about that.
Just
but
through
the
it.
disc and stuff.
Okay.
So you did sort of, it makes sense that the permeability would be less the higher the loading magnitude.
Mhm.
But then you also said that when you induce when you have more variation in your other experiments, you saw increased creep or increased. You saw it was the vertebral body lost more height, or the spinal unit lost.
Right.
So if higher variation was associated with increased reduced permeability, wouldn't you expect to see sort of less reduction in the height of the spike?
I think initially in a healthy state, yes, but as damage accumulates, like we saw in those pores, so the pores, there was no reduction in the pore size, that it would create more cracks into the end plate and therefore allow more fluid to leave. Does that make sense?
Say it again.
What did I say? No, I'm kidding. So I think initially, yes, but I think because there's likely to be a greater accelerated damage, that those cracks would almost in effect increase their permeability acutely. But those cracks over time are also associated with a long and complex cascade of ossification, which over time reduces the overall permeability, which is a degenerative disorder that I haven't gotten to.
Very complicated.
It is. We ended with way more questions than we started with.
Well, it's just too many tissues to understand.
Yeah.
What's going on.
Can you, sorry, go ahead. Okay, I have a basic question. So, are there any other regular material? I don't do this stuff at all.
Sure. Yeah.
So are there any regular materials that show a similar relationship between load variation and increased failure or deformation? So I did have a conceptual model when I think about more complex like bone, like steel. I don't know anything like steel or anything. They not show variations in construction.
So to my knowledge, I don't know of anyone who's introduced the random variation into the force control. We were the first to do it in the spine, but I don't know how, if it's been done in like independent tissues like bone. To my understanding, it's all to the same magnitude using a sine wave or a Haber sine wave.
I would just done mechanical testing.
Not that I know of. It could. I don't know.
Jackie, do you have an idea, like what's that kind of recovery do you expect you would introduce, kind of like interrupted cyclical load? Obviously, sounds like a lot of damage that you showed in those fluorescence ones. I assume you're not recovering from that stuff, so this is more just
no. And so that's been a question that has been I've really become interested in, because this is an in vitro model. So the recovery, like we can only get aspects of mechanical recovery here, but I'm more or less so interested in some of that biological or metabolic recovery that may improve the tissue strength. So I don't know. We haven't done that in a live model, and that's somewhere I hope to go.
Yeah, and I guess I wonder, like with this variation stuff, sure the high variability looks bad, but you could also make an argument like in bone, it seems that obviously loading bone at a high magnitude would be detrimental, but can be really positive for adaptation.
And it may be. That's an interesting question. Yeah.
So do you test at all whether or not you have valuable cells in your preparation? Or are your cells dead? Like, how do you get the preparation? Do you keep it for a long time before you test it, or?
Like the fluorescence testing?
Yeah.
No, it was done right after.
So these are fresh specimens.
Fresh frozen.
Fresh frozen.
Yeah.
And you didn't.
So I would assume the cells were
You didn't
dead.
test for cell viability.
No, we did not do that.
The other question I had about that, since you mentioned that you measure creep, do you ever reach like a steady state where, so creep kind of stops, you reach your steady state, or does it become smaller and smaller and smaller throughout the entire 5,000 cycles?
It usually becomes smaller and smaller and smaller. Yeah.
So presumably there will still be some fluid pressure there.
Yeah, and like when we transect it open, it's not like there's no fluid in there at all. It's just presumably a lot less than what we started with.
I was just wondering, like the cartilage of these, that we treat as a photoelastic material as well. It's been fairly well shown that initially when you load, it's kind of all the water bears all the load, and then you repeat the loading, and then less and less is.
And then the collagen bears it.
More and more on the matrix. And so
Yeah.
I was just wondering if you have tried to estimate that, because presumably the matrix gets injured, not the water.
Yeah.
So, and as you do more and more cycles, there will be more and more loading on the matrix, even though your external load is exactly the same.
That's actually a point that we had in our discussion of that paper, but we didn't explicitly test it. Yeah.
I mean, yeah, I'm not sure how you would test it.
Oh, okay.
I guess you could measure the pressure, I guess.
You could maybe the intradiscal pressure.
Yeah, or you could model it. I wonder if anybody had a, trying to model how the fluid flow would go and how much the change from fluid flow load would be to matrix flow load.
Can you talk a little bit more about these node things?
Sure, yeah.
It just kind of looks like what you see on a histological slice, subchondral bone at the knee, kind of, especially in an osteoarthritic state. So
Yeah,
the
so
green is the subchondral bone,
correct, yeah.
and the orange is the interface above. It's the
Cartilage,
cartilage.
Yeah.
It's fibrocartilage, right?
hyaline.
It's hyaline cartilage.
Yeah.
Okay.
Yeah.
All right.
So
That's the end plate.
that's the end plate. So that's the cartilage end plate.
Okay.
So there's very competing hypotheses of why these types of nodes develop. There's a strong clinical group that believes that they're due to traumas. And there's others that believe that it's due to damage to the subchondral bone first, or necrosis or fragility that occurs in the supporting subchondral bone. And so unlike in neutral, where there was actually no node formation, and this is likely because the load distribution was more evenly distributed across the surface, but in flexed, we may have had more concentrated forces in the central region, which may create some subsurface damage to the bone, and which may start the initiation of this node pattern.
Yeah, it's interesting because it just doesn't look like damage. It looks like a remodeling unit went through that bone, carved a hole, but it really didn't happen that way, right?
Yeah.
This is all ex vivo, so.
So Jackie, what's the cartilage supposed to do? I mean, it's like 50 to 100 microns thick. It's very, very, very thin.
Mm-hmm.
So what do people say the cartilage does?
Like its function?
Yeah.
So its primary function has a lot to do with, because the disc is avascular, and it's porous, it allows fluid to flow through the disc, and it's believed to be a largely load absorption in the spinal unit as well, because it deflects as compression is applied through the spinal column.
I just would have assumed it would be much thicker. I only look at the numbers now, and I thought, this would be half a millimeter or a millimeter thick, and I'm really surprised to see that it's 50 or 100 microns. That's like.
So in the comparative work that I did in study 1, that was one of the differences we saw between the porcine and the young human end plate. The aged human end plates were very thin and very degenerated.
Also very young humans.
Yeah, so I did this same comparative test on the porcine cervical spine, so this, as well as the young human spine, so below age 33, so we had multiple donors, as well as the age spine, so over 83 was our demographic there. So it is thicker in the young human spine.
So you have this Poisson's effect, I think, from the material mismatch, and then you say mode 1. So you basically pulling the subchondral bone apart.
So we have similar questions, and we're actually working with Dr. Cronin with some of his finite element work to understand how, what fails first. And so I think inflection, it has to be some kind of tension force that's pulling it apart. But yeah, it's a question we've had too, and we're running simulations with their spine finite element models right now.
In humans, at night, at least at night time, we know that the disc rehydrates, because first thing in the morning you're about 2 centimeters tall than you are at the end of the day.
Yep.
do we know something about the time constant of loss of fluid from the disc in compression and the time constant in sucking it back up again? Is it a short period of time? Is it an hour? Is it milliseconds? I have no feeling, because I keep asking the question in these compression cycles, if they're sinusoidal, as opposed to changing the duty cycle. So you in a living tissue, you put a compression, and then your duty cycle is a little relaxation time, and maybe if the time constant is such that fluid can come back in again, and there will be some time at which the next compression you're starting from zero, and technically you can go forever. And so those compressions where the sinusoidal and doing 3,000 or 5,000 is somewhat, particularly in a non-living tissue. Are you looking at something that doesn't happen physiologically? If that time constant to read high, so my question is, do we know the time constant for bringing liquid back into it?
So if we just set the spinal unit in a bathroom without
in a physical, no, not that.
Oh, so
In a
physiological?
System, if you compress and then let it go, how long to come back up? Is it half a second? Is it half an hour?
I don't know. I don't know the number value of that. I don't think there's any data on that, but it's an interesting question.
Thank you for your presentation. So I just have out of curiosity. So when people model lumbar spine, usually they assume that the center of the nucleus pulsus is at the center of the endplate. So when you did the indentation sequence, so I was wondering if you noticed that the center of nucleus pulsus is always at the center of the endplate, or during some samples, you notice the some variation.
There is some variation in terms of like I wouldn't say it's always in the center in terms of the medial lateral direction. There is some variation in terms of where it sits anteriorly and posteriorly. It's not a lot. The central region is also associated with, it's the thinnest and most porous, and so it's the region that interfaces with the nucleus pulposus.
So which direction, anteriorly or posteriorly, you notice the most
It's more posteriorly. Yeah.
So one of the results that I was surprised that maybe you're not, is that, for the zero cycles in the flexed and neutral position, you got a similar ultimate compression load there. Did you expect that? Is that known, or was it a surprise?
So this will be a result of how we did the ultimate compression testing. So I mentioned that we only positioned the spinal units in flexion or neutral during the cyclic test, but during all ultimate tests, we tested them in pure compression. Yeah.
That makes sense there.
Yeah.
Well, this is all, like isolated specimen.
Yeah.
What do people think muscles and ligaments that make up the spine will potentially change in the results that you might have?
So I think the muscles will have, I think posture of the lumbar spine will have a greater effect on the effect of muscles. So as the spine flexes, the line of action of the extensor muscles becomes more parallel to the compression axis. So the moment arm decreases and it causes greater compression. So I think the effect of the muscles on the load application is highly dependent on the posture in which the joint is in.
I read once when people use more than just one spinal unit, but
Multiple.
That is a very unstable scenario.
Very.
And as soon as you add some springs that represent muscles, that becomes terribly much more stable.
Yeah.
But I guess that would not really play a role in a single specimen.
No. No.
The other thing that I was curious about is just very simply how do you get that value? Because you said the UCT, the ultimate compression
Yep.
tolerance, then you calculate
Yep.
kind of theoretically.
Yep.
What factors go into that calculation?
Great question. So it's based. So the calculation is a regression equation that's been published, and it's one of Jack's former students who published it in 2005. And in that study, they compared the predictive capacity if they included the end plate area, so the surface area of the exposed end plates, and then you average that, which goes into the equation, as well as they did some measurements if bone mineral density changed the predictions and the accuracy of those predictions. And so the bone mineral density within the porcine model didn't change those predictions. And so the only factor that we measure and that goes into that regression equation is the end plate surface area, so the area of the load applied.
And the only other thing that I was a bit curious about.
Sure.
Was you mentioned this one study that somebody did where they measured the cartilage tensile
Yeah.
loads. Why would you not look at compressive loading and tensile loading in cartilage? Seems to be a strength.
So in the end plate, especially in the caudal end plate, it's a concave surface. And so if you assume the pressure distribution from the nucleus pulposus is even, it's like a suspension bridge. So it's going to put the cartilage in tension. Yeah. Yeah.
That's all the time we have.
Perfect.
So let's thank our speaker again.